before they start to tell us a little bit about what they do in their research. Numbers still going up, 234, amazing. We're stabilizing a little bit. Just give it one more. Still going up at quite a rate. Matthew, just while we're waiting for people to join, can I check, is it your good self doing the single maths tutorial this evening?
Yes, it's me doing the single maths.
Okay.
I figured I'd keep an eye on how this one finishes to know if they need extra time for a comfort break or whatever before I get started on the next thing. That's
perfect, thank you. I suddenly realised there was logic there. Okay, we're at 285, and I think given the time, we are going to make a start. So welcome to everybody that is joining us this evening. Delighted to see so many of you here for what is going to be a very interesting and exciting talk. It's my pleasure to introduce Professor Ray Goldstein, a colleague at Churchill College in Cambridge as well. Very good to name drop the college in this process at the same time. But is Professor of Mathematics in Cambridge and runs a research group that spans many interdisciplinary aspects of mathematics, of physics, and colleagues in all different fields, which is very apposite for this STEM Smart course, where you're all studying different subjects in chemistry, biology, physics, and mathematics. I don't know how bored Ray gets of people mentioning this, but one of Ray's claim to fame is that he has won an Ig Nobel Prize in physics. Was it in physics or in maths, Ray? In physics. In physics. So I'm sure if you have questions about that, Ray would be happy to answer those as well. As per usual in our tutorials, we are going to use the Q&A to ask Ray any questions that you would like to at the end. We will manage those, and we will be asking questions at the end. So if you do have any burning questions as Ray goes through the material, then please make a note of those, type them in the Q&A, and we will make sure that we group those together towards the end. We aim to finish at between 5:45 and 5:50, but as you can see, your single maths tutor is here and will take into consideration what time we finish today. So as I say, I'm delighted to welcome Ray. We are very honoured and privileged to have him here this evening. And so I'm going to hand over now for him to tell us what he's going to talk about. Thanks, Ray.
Well, thank you very much, Lisa, and good evening to all of you. This being a Zoom session, I don't see any of you, so I'm just trusting Lisa when she tells me there are 280 people out there. Otherwise, I'm delighted to see you. When Lisa asked me to give this talk, I scratched my head a bit to figure out what would be the appropriate topic. And I settled on one, which is the title of today's talk, How Microscopic Organisms Turn Themselves Inside Out, because it's a talk about a project that involves physics and math and biology in a very interesting way that I hope you'll find entertaining, maybe a bit amusing, but provocative scientifically. Now you might ask, why should we study such a thing? Why is this a subject in science? And I'm just going to start with a quote from a very famous developmental biologist. And I should say, though, that you'll notice I'm not quite in presentation mode here. We discovered Zoom is having a little bit of hiccups with that. So we've made it look as close as it can be. So here's the place to begin. The famous developmental biologist Lewis Wolpert said once, "It is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life." Now, you can't find a paper in the scientific literature where Lewis Wolpert said this. He said it to a doctor as he was having a discussion with him, and it was written down by a friend who was party to the conversation. So what was Lewis Wolpert actually talking about? So gastrulation is a process that occurs in early embryonic development in all of us, animals like us and down to the simplest kind, have the following process that goes on in early development. So after the egg is fertilized and there have been a number of rounds of cell division, you end up with a structure that is basically a hollow shell called the blastula. And in this cross-section, you can see it's basically a single layer of cells on the outside with essentially a fluid-filled interior. And over the course of the next epoch in development, this structure develops a kind of invagination, a fold inward, which eventually produces the gastric system, hence the word gastrulation, and the gut that we all have. And in fact, eventually, what was a sphere, topologically a sphere, ends up being a torus in the sense of having a hole all the way through, as we all do from our mouth to the other end. So this is a really fascinating process. And what Wolpert was basically saying is that if in the early stages of development, this doesn't happen, well, you're not here. So it's a lot more important than almost anything else in your life. And this kind of geometric topological rearrangement of tissues is something which is not only central to biological development, but it's also the subject of a tremendous amount of research historically, of course, but also in the present age when we have great visualization tools to actually see what's going on. Now, one of the things that as physicists looking at problems in biology is always at the back of our mind is that we would like to examine these kinds of processes, but we'd like to have the simplest possible system to study it, to see if we can make some progress in quantifying and understanding what's going on, particularly from a mathematical and physical point of view. So while it's true that studying animal embryos is probably the way to go, it's complicated for many reasons, some of which are probably quite obvious to you. So the question then is, what kind of organism could we study where we could see these sort of tissue folding and topological rearrangements in a way that we might be able to quantify more easily? Now, it turns out to the rescue comes one Antony van Leeuwenhoek. So I don't know how many of you have ever heard the name of van Leeuwenhoek. He was not the inventor of the microscope, but he was sort of the perfecter of the microscope, working in the late 1700s. And this is a picture of him, but it's also a picture of a replica of his microscope. I actually have one sitting on my office shelf that I bought in a museum in the Netherlands. It's a tiny little device made of brass with a tiny little millimetric lens, which he was a magician at making, that had a resolution that was vastly better than any other microscope of the time. And it had a little sort of translation stage, and you'd put your sample at the tip of this pin, and you'd hold this up to the light, maybe to the not directly to the sunlight, but to the bright sky, and then you could see amazing things. And he discovered a whole bunch of really amazing things in biology and actually revolutionized the whole world of biology through his discoveries. And as he went along in science, he wrote many letters to the Royal Society of London, which was at the time, the premier sort of new scientific establishment that was making great advances in the way that science was done. And here's one of the letters that he wrote. You can get this digitally online. It's a letter from Mr. Antony van Leeuwenhoek concerning the worms in sheep's livers, gnats, and animalcula, that was the name for tiny little organisms, in the excrements of frogs. So it may make you wince a bit to read a title like that, but this man was fascinated by what he found basically in fresh water. In fact, if I, sitting in Cambridge, go down to the River Cam and I scoop up some of that water, I will find the very things that van Leeuwenhoek found. And one of those organisms that he found is shown in his figure 5 up on the upper right here. This is in the year 1700. And it's a spherical organism with lots of green dots on it and smaller spheres on the inside. And if you read the wonderful Old English here, he talks about how it moves in an amazing way. And these smaller spheres somehow exit the larger sphere and apparently are the next generation of this organism. But the organism remained unnamed until almost 60 years later, when Linnaeus, the namer of almost everything in biology, in his great work on taxonomy, in the very last entry in the whole book, gave it the name volvox, from the Latin root volvere, which means to roll or spin, because as this organism moves around through the fluid, it actually spins in a very characteristic way. And if you read the Latin down here, it basically says that it appears to rotate without pause, not P-A-U-S-E, but P-A-W-S, like pause, hands that would actually make it rotate. But we now understand that this organism rotates through the action of microscopic little hair-like appendages on every cell that push fluid around very much the way they do in our respiratory system. So for you to understand exactly what we're talking about here, here's a modern video we took in the lab and that showed up in a BBC documentary a few years ago. It may play a little bit jittery, but I hope you can get the idea. Here's this organism about 0.5 millimeter across. The little dots that you see on its surface are each individual cells with about 10 microns in size, and each of them has these tiny little hair-like appendages called flagella, and they make the whole organism spin as it swims, and that's what Van Leeuwenhoek saw, and which gave it its name. And if you look carefully where my cursor is, the smaller spheres on the inside are actually the next generation, the daughters of this mother, and the dark green dots within those daughters are the next generation, the grandchildren, so to speak. And this organism is a model organism in biology for many interesting processes, and I'm going to tell you a story today about how we've been looking at one of those processes. And just to put this in perspective for those of you who've learned about evolutionary biology, this organism Volvox, which I show you on the right here, and which is again some decent fraction of a millimeter across, half a millimeter across or so, is a sort of descendant, cousin maybe, but more like a descendant of a single cell organism called Chlamydomonas, known as Clammy to its friends, which is only 10 microns across. Remember, your hair, individual hair follicles are about 100 microns across. So this is a really tiny organism. And here you see these two flagella that allow it to spin. So this is conceptually like each of the dots on the surface of this sphere. And that allows it to spin through its fluid environment and do many of the things that Van Leeuwenhoek saw. Now, as Lisa says, I'm a professor in mathematics. And in fact, this is what the Cambridge Mathematics Department, the Center for Mathematics, looks like. We have both pure math and applied math here. And it's not normally the place you'd imagine to find a bunch of people talking about green algae, but this is one of the virtues of mathematics in the UK. It's a very broad church that has many different kinds of science that go on within it. And so in fact, in the basement of this wonderful facility that we're in, you can find my lab. And in my lab, you will find something that looks pretty much like a biology department. So here are some snapshots from different areas in my lab. Here are some of my associates working on growing some organisms, actually green algae. Here are some of the chambers in which we grow these things. These are photosynthetic organisms. They need a day-night cycle, just like plants do. And so these big boxes have a temperature and day-night cycle to them. And if you look inside, you see flasks full of green algae growing up inside of water. And we have all sorts of fancy microscopes, like this one here, that has a fancy high-speed camera to study the dynamics of these things. And we even build our own kinds of special microscopes, which I'll tell you about later, to study this process. So it's a math department, yes, but it's got a working cell biology lab. And many of the people in my group are actually biologists. Okay, so the other thing is that when you want to do these experiments, you not only grow up the organisms and prepare them to put under the microscope, but sometimes you even need to be able to keep them growing while they're on the microscope. And here's a little device we've recently been working on. It looks like some crazy science fiction contraption, but it's a whole bunch of sensors and controllers to control the temperature and humidity inside a little box that sits on the microscope, so that when we do these experiments, we keep them happy. The organisms can grow and divide in their usual way. Okay, so let me tell you one more little piece of biology here that's kind of interesting. These organisms have a very exciting 2-day life cycle. And if you pick it up, as indicated here, between light and dark, in the morning of the first day, kind of like in Genesis in the Bible, you have a mother that has these daughter colonies in it, shown schematically as these spheres with the green reproductive cells. And the first thing happens is that the children are born. So basically these smaller juveniles exit the mother, and they're now sitting there with their reproductive cells. And during the first day, these cells grow very, very large, and then rapidly go through 9 or 10 rounds of binary division, producing 2 to the 9 or 2 to the 10 cells of the daughter colony that will be the next generation. And then overnight, an amazing thing happens, because these little spherical daughter colonies, which have to look like the mother eventually, are actually produced inside out. So they should have their flagella pointing out so that they can swim through their fluid environment, but they're actually pointing inward. And this mistake, if you want to call it that, has to be fixed. And the way it's fixed is that these little spheres actually poke a hole in themselves and turn themselves inside out, as you see through this brief set of snapshots from electron microscope studies, where you can see this hole opening up, enlarging, and then the whole thing flipping around. And this is called embryonic inversion. And it's essentially like the gastrulation that I told you about happened in all of us when we were just a few cells. And then the rest of the whole life cycle just adds a kind of matrix that holds the whole cells together in a way that inflates this balloon, but there's no more cell division or anything like that. Now, it's known historically that there are two types of this turning yourself inside out. And here are sort of crude videos taken by standard microscopy, where you can see that hole open up, and then the lips peel back, and the whole thing passes through the center, and you end up with the inverted colony. And once it's inverted, it's flagella operated, and it starts spinning. And schematically, what you have is a kind of cross-shaped opening. These four lips peel back. The thing then thrusts this anterior through, and there you go, you've inverted it. But there's another version, which is really like gastrulation, which is shown here. There's this kind of rim around the center that contracts, and then the whole thing just literally turns itself inside out with no assistance from anybody. And when I first saw this, I said, we really have to understand this. We have to understand the mechanics of how this works, the timescales. It takes about an hour. And so it's a convenient timescale to study under the microscope. Now, you might ask, how on earth can an object just sitting there by itself turn itself inside out? And the clue to how this occurs was achieved by studies with electron microscopy by Stephanie Hoon, who is a postdoc in my group, but this was done in her PhD thesis in Germany with Armin Hallmann. And so they managed to stop this process at various stages biochemically, and then put these structures under the electron microscope, where you could get a very high resolution snapshot of what was going on. And roughly speaking, it looks like a sort of mushroom. So here's it at an early stage from several different views. And here's a cross section of that shape. So it started out being a sphere, and its cross section would have been a circle. And now you see it's got a kind of hemispherical cap and another hemispherical base, and it's connected by this funny curled region, which extends all the way around. And that curled region moves up, and then the outer part spreads around and reconnects. And the way in which this bending occurs here is quite miraculous. It involves the change of shape of the cells and the way in which they're connected to each other. So in fact, if you look very closely in this region, what you can determine is that if I show the cells in green here with their nuclei in blue, that there are little filamentary connections between adjacent cells, shown as this continuous red band. And those connections actually move from the center of the cells down to the tip. And so imagine you had some bowling pins and you connected them together with elastic bands in the center. Well, they'll just be straight like this. But if you cinch them together at the tips, they're going to flare. And if they flare, they're going to try to bend the sheet that they're in. And so the cells spontaneously change shape, and that produces this bending that ultimately produces the entire shape change. So it's a process of shape changes and cell connections all working together to invert this sphere. So if that's the microscopic physics, how is this exactly working? Well, basically, if you look at a cross-section of a cell, there are long filaments through the cell. They're kind of like railroad tracks along which proteins move. These filaments are called microtubules, shown in green. And the cross connection between the cells is anchored on little proteins that literally walk along these filaments and carry them to one end, as you see here. And we know this because the biologist went in and basically stopped these proteins from working, and the whole process stopped. So it's a microscopically very complicated process, but it involves these concerted motions of cell shape changes and connectivity changes. Okay, so now, how on earth can we study this in a way that gives us a three-dimensional view of what's going on? And it turns out that there's a technique that's been developed over the last maybe 20 years or so that is known by various words. One of them is selective plane illumination microscopy, SPIM, or light sheet microscopy. And the basic idea is quite similar to the way you might take images in a magnetic resonance imaging device, if you ever had one, if you broke your leg or something like that. MRIs take slices and build up a three-dimensional view by building slices of the world and then assembling those pictures. And so the basic idea here is that if you have a laser from far away coming through a lens, which is basically a microscope objective, but it's going through the opposite way than the usual way, you'll take a large sheet of light and turn it into a very narrow sheet of light. And if you send that through your sample, as you see here, then if the sample gives off light in response to the laser illumination, which chlorophyll does, then the light going through here can be imaged by a camera. And you basically can take a series of images by moving this suspension of the organism through the sheet and build up a stack of images that way. And so optically, what we have here for people who are maybe on the physics side is you take a laser from far away, you split it into two paths with a beam splitter, and then you send each of those beams through a lens. So it takes a very narrow beam and turns it into a wide cylindrical beam. And then you send it through a cylindrical lens, and that turns a round beam into a sheet, a line. And then you send it backwards through the two microscope objectives, and you can illuminate from the two sides. And here's your camera. So here's what it looks like. It all fits on a tabletop. So here's a camera. This is the simple version where there's only one light path. The laser comes from below, goes through some optics, goes through an objective lens backwards, and there it is in the chamber. And if you look more closely, you can see here two microscope objectives in the simplest case. So the light's coming in, and then the fluorescence light is coming back out to a camera. And if you do this more professionally, and you actually have two light sheets coming in, then you have a chamber where there are three microscope objectives, all meeting very close together, all engineered to work precisely. So the sheet can come in from this side, the sheet can come in from this side, and the light to the camera goes out that way. Okay, so it's quite an amazing situation. You can view mostly transparent samples of sizes reaching several millimeters, but get incredibly fine resolution. The sheet thickness is a few microns, a few times the wavelength of light. And so you can actually get incredible resolution. So here's what happens now. If you build up an image of the entire colony by taking a series of slices at positions that are stepping through this large spherical colony. So when you're just near one edge, you're just going to see a small patch, a circular patch, and then eventually you're just going to see the edge. And if you step through slowly, I play this video. Sorry. If you step through slowly, there you see. So here you were slicing through the daughter colonies on the inside, and here are the cells on the outside. And as we get to the other end of the spheroid, then you see it collapses back to a nice little hexagonal pattern right there. So those are the slices through a sphere. And now, if you think about it, the way your doctor will assemble the slices in a magnetic resonance image to get a three-dimensional view of your brain or your arm or whatever, we can do the same. So we can actually build up a video, a three-dimensional video, maybe four-dimensional, 3 space plus time, where we can watch the inversion process of a whole bunch of these embryos all at once. And this was such a striking video and story that it actually made it into the New York Times with this amazing title, Birth Trivial Next to Cellular Donut Making Process. I like that title. So here, this is, again, note the time stamp down here. So here's the mother. It's got a whole bunch of these daughter colonies, and they keep going through this amazing process. And once they flip around, they actually start rotating because their flagella are now facing out the right way, and they can swim. So it's quite an amazing process. Because it's sped up like this, they seem to swim ridiculously fast. But it's possible then to look more precisely at any one of these processes. And here you can see now embryonic inversion in all its glory happening over about 45 minutes. So there are all these strange little fluctuations that occur. And eventually there's a concerted motion where there's this kind of ring of contraction. And the whole thing wraps around and closes up. So it's really quite amazing. And it does it all by itself. It's genetically programmed to turn itself inside out. Now we can study many of these because we're patient, and we can look at many of these processes. Here's another one. And again, with this view, you can see again this contracting ring. And then there's a hole that opens up at the top, and the whole thing then stretches around and reconnects and starts rotating. So it's really quite extraordinary to see. If you're very patient and very fortunate and clever, you can actually see this with a technique called confocal imaging, which has a bit higher resolution, but it can't quite image as deep into the sample. So it's not quite a view of the entire thing, but I think you'll agree that this is quite a striking view. So here we're looking down the sort of axis of the whole thing. And just where my cursor is this little hole that is kind of pre-patterned there. And that's the one that will open up and stretch. So watch this video. There are, again, fluctuations. Each one of those green dots is a single cell. And so there's a bit of contraction, there's a bit of fluctuation, and then the whole thing expands and then turns around and contracts. So that's what we're after. How on earth are we supposed to think about this process? How on earth are we supposed to measure anything and turn it into something we might think about physically? Well, there's some obvious things you could do. These structures have a kind of symmetry to them. You can draw an axis through the center, and roughly speaking, they're surfaces of revolution around that axis. So here's that central axis. And I can look at the radius as a function of, say, the angle from the top, or maybe as a function of the arc length down from the bottom as I move along this surface. I can measure the local curvature, 1 over the radius of curvature of a circle I could inscribe in here, and I can color code the surface by that curvature. And of course, you see regions here with very high curvature shown in red. I could measure, for instance, the distance between this bend region and the bottom as some measure of how far along this process has gone toward inversion. And what you discover is, as in many cases in biology, there's a lot of noise, there's a lot of variation from one organism to the next, but there is a kind of commonality. So here I look at this position E, which is just normalized by its initial value, as a function of time, where we've shifted time to match up the halfway points. And you see that there's a well-defined smooth variation of this. There's nothing quite abrupt going on. It's just slowly being flipped through. And in fact, that's kind of interesting, because if you took a tennis ball and cut it in half and tried to force it through, you might find, depending on how you do it, that there's a point at which it sort of runs away, kind of just goes mechanically unstable and flips. That's called a snap-through instability. But we don't see any evidence of that here. You can go around and measure the surface area of the whole object. We just use the geometry of the shape and can measure the surface area. And what you discover is that there's actually a large expansion, and then there's a very large contraction. So not only can this cell sheet that we're talking about bend by virtue of cell shape changes, but it can expand and it can contract, because cells can get short and tall, squat and tall, or they can get fat and wide, and that can change the shape and geometry of the cell sheets as well. You can basically measure the size of this opening. It's basically not there at all, and it suddenly snaps wide open and closes very fast. So there's a lot of elasticity to this process. So in thinking about how to describe this mathematically, we were motivated by something which is inside all of you. So if you were to look at the red blood cells that circulate in your veins and arteries, with a very high power microscope, such as an electron microscope, you would see that they look like a kind of deflated sphere. It's a shape which is technically called a biconcave discoid. It's like a bagel, but doesn't have a hole in it. And this funny shape is very important in the functioning of red blood cells. And for many years back in the 1960s and 70s, there was a lot of work in trying to understand the mechanical origin of this shape. And if you think about a football that has been deflated partially, where there's a dimple somewhere in it, you could appreciate that in some ways it might be that this shape is sort of a compromise between the stiffness of the membrane, which basically wants it to remain sort of flat, or as flat as it can be, and the volume constraint of having a smaller enclosed volume than the equivalent sphere. And a physicist named Wolfgang Helfrich speculated, and has now been shown to be correct, that we can think about the energy of this surface in a way which may not be something you've seen yet in your math classes, but I think you can appreciate, in a way that's characterized by its local so-called mean curvature. So everywhere on a surface like this one, I could draw two little circular shapes which would characterize the local radius of curvature at that point. So those circles would be designed to have exactly the same shape, the same curvature as the surface at a given point, and would just tangentially meet it at that point. Now, in the center here, there are two orthogonal directions I could go in, and they have the same curvature. But in other areas, there'd be different shapes in two directions, such as up here. And the quantity called the mean curvature, which is the average of 1 over the first radius plus 1 over the second, is a measure of how bent the shape is. There's a second quantity that is called the Gaussian curvature, which we don't really have to talk about, but it's the product of the two curvatures. And Helfrich basically said that the energy of this surface is an integral over the surface of something that looks like the curvature minus some constant squared plus possibly some other piece. But this is the piece we should focus on. So this second constant is sometimes called the spontaneous curvature. It's a statement, if it exists, that the surface doesn't really want to be flat. It wants to be bent with a certain shape. And your tennis ball is a good example of that. If you cut it up into pieces, those pieces just stay bent. So their happy configuration, their energy ground state, is bent at some curvature. And basically you pay an energy cost quadratically in the difference between your curvature and the preferred one. And if you take that kind of energy and then you calculate through a complicated calculation what the shape would be if you deflated it, you get exactly this shape. So that's a kind of plausible starting point for the discussion of these problems, that the effect of these cell shape changes is actually to make a preferred curvature which tries to bend the surface. Now, in biology, there are many places where you find preferred curvature. And here's just an illustration. If you take a membrane, which in cross-section looks like this, the kind of membrane that is the boundary of your cells, there can be difference between the molecules on the outer side and the inner side, which can lead to some preferred curvature. You could have proteins that stick to the outside and not the inside, or insert themselves on one side or another and crowd around the outside. This is well known in cellular biology as a way to impose a preferred curvature on the shape. But actually, all of you are in possession of objects with preferred curvature because you have, I think most of you, I can't see, most of you have hair. And that is one of my favorite subjects outside of cell biology. So here are some pictures from a real human ponytail of hairs, just randomly selected from such a ponytail. And this was part of the subject that won that Ig Nobel Prize some years ago. These have been digitally fattened up so they're easier to see their thickness. But what you see is that they have all sorts of different shapes, and they're isolated. They're just hanging from a support. They're not interacting with each other. Their natural shape has all sorts of bends and curls, and that, you know that, right? That's lesson 0 in possessing hair. And so that shows you that there can be this idea that the actual preferred shape of an elastic object is not straight, but it's bent with some spatially varying preferred shape. So it's one thing to be a filament like this and to say, well, I have these preferred shapes. It's pretty easy to adopt a shape which is everywhere the preferred one. But if you put that on a sphere, it's not so easy. And so I'm going to give you one slide to give you a flavor, and I won't quiz you on this, I promise, but one slide to give you a flavor of how mathematically you try to encode this into a proper theory of the elastic properties of a surface, and then I'll skip the math, right? So the proper theory looks something like this. You say, I have a reference shape where I know what's going on. That's on the left. Say it's a sphere. On the right, you have some deformed shape, and I'm essentially going to make a map from every point on my reference shape to my new deformed shape. And it has, the reference shape has its arc length variable S0. It has, the new shape has its arc length variable S. And I can use some geometry to express, for instance, the radius and arc length and how they're differentially related to each other, and various geometric quantities like the angle that the tangent to the surface makes with respect to some axis. And the most important point here is how I use these ideas to formulate the energy. And the whole point of this description of a surface is that two things can happen. It can stretch, just like a piece of rubber, or it can bend. And if you think about stretching, well, I can have a spring, and the energy to stretch a spring goes like 1/2 kx squared, where x is the displacement. But if the rest length of the spring is non-zero, then it's 1/2 kx minus x0 squared. And that's essentially what you do. You construct things that are called strains, and these strains are related to the material stretching in one direction or another. And they differ from some preferred value, call it Fs0 or F(phi0) in the two directions. And the energy looks like a bunch of squares of pieces like Hooke's law for the stretching part, and another piece that involves essentially the bending pieces I told you about before that have these functions K, which are like curvatures. So the problem for the reader is, given an energy that looks like this, where it's a surface integral with some prefactor that measures the stiffness, etc., find the minima. And this is a very hard problem mathematically, but this is what computers were invented for. So I'm going to show you what happens if you take a spherically symmetric surface, which I'm going to look at just in cross-section, but you should understand that you could just rotate what I'm showing you around and you get the full shape. If you endow that shape with a little region in red initially, which wants to bend inward because the cells have changed shape and they're imposing an inward curvature. And I'm going over time in this video to dial up the region over which that is occurring and the strength of that preferred curvature. And you'll see that if that's all I do, just dial up the strength of that bend, then I will produce on the left here something that you might call the purse string effect. So if all that the cells did was to contract in that region such as to produce a bend, it would get stuck. You couldn't possibly take this bottom part and stick it through the top part, which you must do to invert. But then if you generalize the problem and allow for what we call posterior contraction, namely that the part of the cell sheet down here can actually have cell shape changes that contract the sheet, and the top of the sheet has cell shape changes that can expand it, then pretty much you can fit the bottom into the top and you can start inverting. And you can test whether this makes sense because you can change the sign of the curvature that you impose here, like you see on the right here, and it doesn't work at all, and that gives you a kind of smiley face. And that's interesting because if you actually look at the actual shapes, you might have looked at this region where my cursor is and said, oh, but that's actually bending the opposite way of this. Maybe that's where the preferred curvature is, but you can't just do that. You can actually rule out and rule in various scenarios. Okay, so if you put the whole thing together and put a little hole at the top to indicate that there is really one in the biological system, then again, these are cross sections through a three-dimensional shape. You can see that as you dial up the bending and then you spread this wave of cell shape changes all the way across the bottom, you can literally turn a sphere inside out, and it looks pretty much exactly like what we see in the experiment. In fact, you can put together the experimental observations shown in black here and average them over a whole bunch of different embryos doing this, and you can fit the parameters of the theory to this kind of process and actually get the entire inversion process captured by a model that's as simple as this. And when you put that all together, this is what it looks like. And we can actually recapitulate our experimental measurement of this little progress variable E that tells us how far things have gone if you do this the right way. So you get some contraction shown here. These are actual experimental shapes. Then you get the inversion or the bending region. Then you get the thrusting of this bottom region up through the top. And there's even some cell-to-cell contact that occurs at some point, shown by this little contact sign. And then the whole thing inverts, and then the process is over. So this shows you that actually from what I think are very simple ideas that have to do with bending, stretching, and curvature of elastic sheets, we can actually start to quantify what's going on in this biological system. So in the last couple of minutes, let me just tell you one other amusing thing that happens. And this is a good example of how or what physics and mathematics can often bring to biology, because we try to take a point of view here, which is, yes, we're interested in understanding what happens in the actual biological system with its parameters the way they are. But sometimes we ask a larger question. If I let those parameters vary all over the place, do I learn something about the sort of larger picture of what's possible? And here's what you can learn. So never mind the math too much here. I showed you that in our initial shape, there was a little red region, I'll say it has a width lambda, where we said the curvature was negative and pushing everything inward. Suppose we let that be a parameter of the problem. And we ask, what changes if I change the size of this region? So that's something that we imagine we can control. And then I'm going to measure a quantity I call D, which is just the distance that the bottom has moved up as a consequence of this process. And we can make a graph. So here is the magnitude of that curvature that we're imposing, and we multiply by the radius of the sphere just to make it a number, a dimensionless number. And here's the thing we measure, D, made dimensionless by the radius of the sphere. And these are curves for different values of lambda. So when lambda is small, it's like what I first showed you. And as lambda gets larger and larger, it's more and more different. And you follow a path in this parameter space for small lambda, which is just some monotonic curve. But as you increase lambda, this curve bends over, eventually reaches a point where it's horizontal. And then below that, you end up with this kind of loopy structure. And what that means is that, remember, K is something I control. This is, what the cell is fixing by changing its cell shape. And so what that means is that a given value of K, there are two different possible shapes that you can have, one of which has a small value of D, and one which has a large value of D. So the small value has just moved a little bit, the large value has moved a lot. It's like there are two coexisting shapes. And in principle, when you see something like this, It reminds you of what happens when a liquid turns into a gas. In your test tube, you could have a liquid and a gas coexisting. This is like two different states of matter coexisting. And you might imagine that there are fluctuations back and forth between the two. And I bring that up because there's this wonderful thing that we and others have observed, which is known technically as denting. So if you watch the process of inversion just before it actually does its thing, you discover there are these huge fluctuations in the shape, these massive panels moving in and out. And then finally, it does this inversion. We think that this motion beforehand is an indication there's a kind of mechanical instability in the system, perhaps similar to what I told you about with the mathematics. Okay, so that's the story I wanted to tell. I hope you've gotten an idea that the world of mathematics and physics can be extended to address problems in biology. It's a combination of technology, it's a combination of mathematical technology, but it's also a question of deciding what's an interesting question. And that's for all of us to decide as a community. I hope you found this interesting. Thank you.
Thank you very much, Ray. I found that absolutely fascinating, and I should declare my, should I say, ignorance at this point. I stopped studying biology at the age of 16. Thank you very much for your introduction about, what these different things are, flagella and all of these things, because these are words, as my biology colleagues know, I have to get them to redefine for me every time. So thank you for that, because that was brilliant in order to be able to follow that. We do have a number of questions in the Q&A.
I'm not sure I can see them. Should I go out of sharing?
Well, I'm going to group them for you.
Oh,
Ray,
you group them.
So
okay, you read them to me. Yeah.
Yeah, I can. And you might want to refer back to slides, I think, because some of them may. I'm going to start at the beginning, which is the first question is gastrulation the key process behind diseases such as Barrett's syndrome?
I'm not a medical doctor. I believe there's something called Barrett's esophagus, which is some problem associated with acid reflux. I don't know Barrett's syndrome precisely. I could quickly Google it and see, but maybe the person could amplify, or maybe I'll quickly do that. So I don't know. We can check.
The person that's asked that question, if you'd like to clarify a little bit more in your in the Q&A, we will follow that one up. So there's a couple of
sorry, I can read it to you. Barrett's esophagus is a condition in which the flat pink lining of the swallowing tube that connects the mouth to the stomach becomes damaged by acid reflux. So I think that there might be some shape changes associated with your problematic esophagus, if that's the case, but it's really due to something more biochemical.
Thank you.
I think.
So there are a few questions here related about, and you sort of started to answer it actually at the very end, about what causes the fluctuations. So what causes the fluctuations, and why does it almost seems spontaneous, this turning inside out? Is there some trigger?
Yeah, so that question is at the heart of not only this problem, but almost every problem in developmental biology you might answer. And it has to do with what in physics we call symmetry breaking, the breaking of symmetry. That is to say, you look at this raft of cells that's there initially before, and that's spherical initially before any of this bending occurs. And as we look at it, we have no way of saying, oh, these are the cells that are going to do it. Although we know where the position is on the whole body, because there's a North Pole and a South Pole. So yes, we know, but there's nothing to tell us looking at the cell. Oh yes, of course, that's one that's going to do this. But yet they do that first. And this happens in almost every problem involving geometric changes in biological systems, that there is some special spot that is the first to go. And there is a very important piece of work by the mathematician Alan Turing in 1952 called The Chemical Basis of Morphogenesis, which laid out a scenario by which this could occur. That is, you could what they call spontaneously break a symmetry. If there's some noise in the system and the dynamics are such, it can naturally produce a pattern. But what happens here is not just a random location of that, it's a very well orchestrated structure. So the prevailing view is that somewhere earlier in the process, and we don't understand it for this, there has been a differentiation among the cells in terms of where they are on the daughter colony, such that they first get this program and first go. But the fact that you see these fluctuations beforehand suggests that this may be a process of mutual elastic communication between them, kind of refining where that will occur. And there's probably a bit of a sort of tug of war going on here, and it gets resolved this way. But that is what I would call hand waving. And most of my colleagues would call that hand waving. We don't actually know. And this is one of the major questions. Where's the information to say, start at the equator and go from there?
Yeah.
So great question.
It is a really, and there's quite a few questions in the Q&A about how the findings in this might apply to other areas of science, like are there things that we've learned from this that we might apply to maybe biomedical engineering or engineering? And I when you were talking about that and the perturbations and the fluctuations, it started me thinking about astrophysics and the fact that to stimulate a collapse under gravity, there has to be a slight variation for that to happen. If things aren't identical and locations have to be slightly different, or there has to be sorry, I sort of
no, I understand completely. So let me kind of go backwards through what you said. So the first thing is, all of these problems that involve some kind of, we call it an instability, some kind of transition to a new state, have in common this issue of where do they start and what is the trigger to start them. In the astrophysical context, or any other context that's not living, what you often have is a less precise, let's say, location or orientation of the instability than you have here, because here you have biological differentiation. The cells that are at the top of this organism are known to be slightly different than the ones that are at the bottom, and that persists all the way to adulthood. Now you can ask, where did that come from? That was an earlier stage of cell division, that some cells are older than others when you go through rounds of cell division. So you have to recognize that in almost any system in which there is an instability, there is this issue of a trigger. In some cases, there's a bias that puts it somewhere. So you wouldn't learn too much astrophysical by going from here, because we already know in the astrophysical context about those issues. But you could learn interesting rules for biomedical applications. For instance, this whole idea that elastic structures can have an intrinsic curvature instead of just wanting to be flat means you could imagine designing materials with maybe a programmable intrinsic curvature, such that you could inflate it and it would adopt some non-trivial shape, and then deflate it and it's back to something small. So there are many ideas around like this, where understanding this kind of mechanics can open up new avenues for engineering as well as understanding, because they're not just developmental biology processes that involve these kind of topological changes. In ordinary physiology, there are these things as well. Membranes can bud off smaller vesicles, and that's a topological rearrangement. So there are lots of things like this that occur. But this kind of folding is everywhere in your body. The development of the optic cup, as it's called, the eye socket, basically, the tissue, is the same kind of inversion as you see here.
There's a few questions about the spinning that occurs at the end, occurs as a result. Does it occur as a result of the inversion, or is there something else that then causes the spinning? So the questions we've got is, what do they gain from spinning? Is it their only way of moving around, perhaps?
So yeah, exactly. I know where the question is going. So let's look again at a movie of Volvox. You can't see the individual flagella there, but each of these roughly 1,000 cells on the surface of each of these colonies has these two hair-like appendages called flagella that allow it to move through its fluid environment, and they're organized on the surface to beat from the anterior to the posterior. So, and there's a slight east-west twist, so it moves forward and it spins. And the spinning is an essential aspect of their lifestyle. These are photosynthetic organisms. They need to steer toward the light, to swim toward the light, to do photosynthesis. And that spinning and flagellar action is intimately related. The next time I see you, I'll give another talk. Intimately related to how they steer. And many different kinds of organisms have this habit of helical swimming. So they need this to survive. And the flagella have to be on the outside. If they're on the inside, you can't go anywhere. I mean, if my arms went into me, I wouldn't be very useful, right? So they have to point out in order to actually propel. But interestingly, they're first produced the wrong way around, where what will be the flagella, they're very short initially, are pointing inward, and you can't get anywhere if you just stir up the fluid inside of you. So they have to point out. Now you might say, well, that sounds weird. Why don't they just develop the right way around? And in reply to that, I would say, well, why don't you develop as a torus from the beginning? And the answer is, it's an accident of evolution. There's a mistake that was made at an earlier stage of evolution before we were us. We were simpler organisms that maybe were fine being the other way around. And eventually it was, eventually evolution fixed that mistake, and we go through gastrulation. So this is the same fixing a mistake.
Brilliant.
Yeah.
Brilliant, thank you. So moving on to sort of, just conscious of the time, moving on to questions of sort of how you got to where you are. What drew you to this topic, sort of from your other mathematical research? A pure accident. So basically, at my prior institution before I moved here in 2006, in about 2003, at the University of Arizona, I essentially got a knock on the door from a biologist across the street who worked in the Department of Ecology and Evolutionary Biology, who knew that both myself and my then colleague, John Kessler, a physicist, were interested and knew about fluid mechanical issues in biology. We had been working on bacterial systems. And they had come across in their biology research some phenomena that seemed to suggest that fluid flow was important for the development of these organisms as we see them. That the flows created by the flagella of the mother had an impact on the development of the daughter colonies inside. And this was really interesting. And I knew nothing about Valvox. I had never heard of it, didn't know the first thing about it. But I listened and I thought, wow, this is really quite fascinating. And eventually I realized that there was an enormous set of interesting questions, interesting to me at least, that had not been addressed from physics and mathematical perspectives, but that these were the ideal organisms to address. And so I pretty much dropped what I was doing and said, we've got to go in this direction. And we've been going in that direction ever since. And it turns out that this organism has taught us an enormous amount. If you go to my website, you can find one of my articles is a review article called Green Algae as Model Organisms for Biological Fluid Dynamics. And basically, you can study the flagella, you can study the swimming, you can study the interaction they have with surfaces, you can study the phototaxis, how they turn toward the light. And it's a big organism. I mean, 0.5 millimeter is huge by microscopic standards. And it's easy to grow, and there are many mutants. And now there are genetically engineered versions that express fluorescent proteins, so you can see various parts more clearly. So it's a kind of all-purpose Swiss Army knife sort of tool to study an enormous range of problems. And that's what is often the case in what they call model organisms in biology. The mouse, the fruit fly, all these organisms are chosen not necessarily because they are the most interesting in exactly what they do, but they're chosen because they're easy to grow, they're easy to study, they can be studied from many different perspectives, they can be genetically engineered, and the community as a whole says, yes, this is the right thing to study, and then they all study it from these different perspectives, and we have a huge encyclopedic knowledge of them. I hope that helps. Thank you. Absolutely. There are, the more questions you answer, the more questions there are, absolutely the research model, right? As soon as you answer a question, 100 more. So, but we will have to finish. So we'll save these questions, and we'll talk to colleagues. And if there are some specific to your particular research, Ray, if you have, I'll send them in an email.
Send them in. I can write some quick replies. Yeah,
that would be brilliant. Thank you. And the final question, which I have to ask, given my introduction, was students are quite keen to hear how you get awarded an Ig Nobel. Is it research that you've just published that somebody finds, or do you sort of flag it to the committee? How does it work?
Wait 1 second. Let me get a prop.
Okay, a prop.
Right. So the issue in question is the human ponytail. This is a real one, but it wasn't. Don't worry, no one was hurt in acquiring this. So the point is, I was contacted by Unilever Research many years ago, would I be interested in working with them on a problem involving the physics of hair? And they gave it a more technical description, but that's what it was. And I had no idea what they were talking about, but it sounded kind of interesting. I had never done any consulting for a company, so I thought, sure, why not? I'll go listen to what you have to say. So I went to Port Sunlight, which is near Liverpool, and I met with a bunch of scientists there, who turned out to be physicists, and one, in fact, who had been getting his PhD from the physics department here in the Cavendish. And it turns out they were trying to understand the properties of collections of hair. And why should Unilever be interested? Well, they're a global manufacturer of personal care products like shampoo and conditioner and all that. And they want to be able to quantify what their products do to hair. And so they thought, well, if we get some basic understanding of the physics of hair, we'll know how it changes when we apply a conditioner or something like that. So we were sitting around a table talking, they were explaining this to me, and I was trying to understand what they had done so far, which was already very interesting. And I was holding this up, and I always try to think, what's the simplest problem you can study so we can try to get an answer? And I held it up and I said, well, look, if we really eventually will claim to understand anything, we ought to be able to just explain the shape of a ponytail. And everyone said, oh yeah, that's a good idea. We should do that. And I said, and by the way, if we do, we'll win an Ig Nobel Prize. And I was joking. I was absolutely joking. But we published this paper in 2012, I guess it was, and it went viral. Daily Mail, everybody covered it. And we didn't do anything except one day, my collaborator, Patrick Warren from Unilever, emailed me and said, guess what? We just won an Ig Nobel Prize. So I have no idea who nominated us. You can't nominate yourself. So it must have been someone who, saw the paper and thought it perfectly fit the bill, where it makes you first laugh and then actually pay attention. So.
Brilliant. Thank you very much for your talk and your time this evening, Ray.
Welcome. Absolutely
fascinated. And thank you to all of the students who have joined us. I think we had a peak of about 340, so wow. Fantastic audience this evening. Just for the students' benefit, your tutorial's due to start at 6, but I think, Matthew, I don't know whether you're going to give 5 minutes grace on that, given the time.
I think we'll give 5 minutes just so that people can fetch a drink or get a comfort break.
Brilliant. Thank you very much. And yeah, have a good evening. Those of you that don't have maths at 6 o'clock, have a good evening. And thanks again, Ray. Look forward to
see you. Thank you again, everyone.
Bye-bye.